Okay, hello everybody. Well, I'm just going to quickly check my microphone. All right, that's better. I had it on a different microphone. Okay, so um, welcome to the Unit 6 review. Now, just like any other time that I've done these, this doesn't replace any help that I can offer you. You know, maybe use this as a refresher. But if there are any questions that stem from this review, or if any questions that I didn't answer, please send me an email, and I can answer a lot of questions on email. We can also have some one-on-one chat time through this Blackboard Ultra virtual classroom. So there shouldn't really be any reason why you're struggling in this course. So reach out for help if you need it. Okay, a very simplistic diagram here on the structure of a nucleotide. Now you'll remember that a nucleotide is the building block of DNA. Nucleotides are made up of three parts. The phosphate molecule and the sugar molecule make up the rails of the DNA ladder, while the nitrogen base makes up the rungs of the DNA ladder. So all DNA molecules pretty much look the same, except for the nitrogen base. And there are four different nitrogen bases. So the DNA molecule is not just a chain of nucleotides, but two chains of nucleotides, both chains connected to each other by way of these nitrogen bases. Now, specifically, the adenine nitrogen base of one nucleotide will be attached to the thymine nitrogen base of the other strand's nucleotide. And the cytosine nitrogen base from one strand will attach to the guanine nitrogen base of the other strand. Each strand of nucleotides is called a nucleic acid, and each strand attaches to each other using these specific combinations of nitrogen bases, and it's called complementarity. So the adenine thymine combo, guanine cytosine combination, that's called complementarity. During S phase of interphase of the cell cycle, the cell has decided it needs to divide. So before it does so, the DNA must replicate. There are 46 strands of DNA in the form of chromatin, and it replicates by a process referred to as semi-conservative replication. It's where we get it's where from one strand we get two strands. Each strand is an identical copy, and each strand is actually a double strand. One strand from the original DNA molecule, and the other one is a newly formed strand that used the old strand as a template. All this occurs in the nucleus before cell division even starts. So it's important to remember that if this were a strand from a newly formed zygote, a strand replicating prior to the zygote undergoing its first mitotic division, any mutation occurring here will be found in every resulting cell of the organism. This slide better illustrates the process of DNA replication. It's an enzyme-mediated process. Don't worry about the 5' and 3' notation, but you'll notice DNA polymerase plays an important role in replication. There's a technology called PCR, means polymerase chain reaction. It's a process that involves the rapid and wholesale replication of DNA. It's also worth keeping in mind that while every cell in a nucleus will have the exact same DNA, not all the genes on the DNA molecule are being expressed all the time. It would not be efficient that a skin cell produce digestive enzymes, since the skin is not really part of the digestive pathway. It would also make no sense having a liver cell produce melanin, since melanin absorbs ultraviolet light to protect the DNA in the nucleus. And, well, light, ultraviolet light or otherwise, won't penetrate the skin to even reach the liver. Nevertheless, the liver cells and the skin cells have these genes. They're just not expressed. Okay, so that was S phase or interphase. Let's set the clock back to gap 1 of interphase. This is where the cell is working at doing its normal job, whatever it may be, making enzymes for digestions, for digestion, making hormones if it's an endocrine gland. Whatever is its purpose is doing its work while in gap 1 of interphase. So let's say it is making an enzyme. Enzymes are proteins. Proteins are made up of amino acids, and the nucleotide code for each amino acid is spelled out in the gene portion of the DNA molecule. So the genetic code spelling out this sequence of nucleotides is in the nucleus. However, the protein-making hardware that will make the enzymes is in the cytoplasm. So the DNA molecule is too large to squeeze out of the pores in the nuclear membrane, so a smaller molecule carrying only the gene code is produced. In a process called transcription. The gene code is transcribed onto another nucleic acid called messenger RNA or mRNA. Now, mRNA is a fraction of the size of the DNA molecule. See, the DNA molecule could have hundreds of thousands of genes. The mRNA transcript is only a single gene long. So when the protein is needed, the enzyme-mediated process starts up to produce this transcript. It squeezes out the nucleus and into the cytoplasm where the protein-making machinery is. So this better shows the process of transcription. The DNA opens up at the gene in question, and RNA nucleotides start binding to one of the DNA strands, creating the mRNA transcript. So translation begins in the cytoplasm when the mRNA transcript binds to the ribosome and feeds between its subunits one codon at a time. Each codon, remember, is a series of 3 mRNA nucleotides that complemented the DNA triplet during transcription. Transfer RNA or tRNA molecules connect to the mRNA codons at positions on the tRNA molecules called anticodons. These are the 3 nucleotides that complement the mRNA codons. Beginning with the initiator or start codon, the tRNA molecules attach to it and the subsequent mRNA codons. A still slide like this does not do this process justice. I strongly recommend you have a look at some of the videos that accompany this in Moodle so that you can see the process working. You need to have just a general overview of the process of transcription and translation. You don't need to know the details. So tRNA molecules carry specific amino acids. The sequence of mRNA codons translate into a specific sequence of amino acids in a process called translation. Process ends when we reach the stop codon or terminator codon, which ends translation. So the resulting polypeptide chain disconnects from the ribosomes and conforms to a specific protein. The exact sequence of amino acids is essential for the protein to conform to the necessary specific shape. One wrong amino acid will make the protein useless. So knowing this central dogma of biology is important. Think about the possibility of questions that you'll want to know all the major steps in protein synthesis. Transcription occurs in the nucleus. An mRNA transcript is formed from a specific gene. Translation occurs in the cytoplasm, where the mRNA transcript attaches to a ribosome and feeds through it, one codon at a time, where tRNA molecules carry specific amino acids, attach their anticodons to mRNA's codon, producing an elongating polypeptide chain. So those steps are important. You'll definitely be expected to convert DNA to mRNA and mRNA to amino acids, and occasionally mRNA to tRNA. So make sure that you remember that no matter what information you are given, this table only converts messenger RNA to amino acids. If you're asked to see what DNA triplets produce which amino acids, first transcribe the DNA into mRNA before looking at this table. Here's a couple of example diploma questions. Notice the DNA strand is paired as though it's a two-stranded molecule. The lower strand is assumed to be the template strand mRNA uses to create its transcript. Now, to confuse you, notice the tRNA molecule is below the mRNA molecule, followed by the list of amino acids. Now, remember, the amino acid table in your data sheet converts messenger RNA codons to amino acids, not transfer RNA. All right, so the letter X on the tRNA molecule is simply the complement of A on the mRNA molecule. But before you say thymine, remember that this is an RNA strand which doesn't have thymine, but rather uracil, the complement adenine. And Y on the mRNA complements the cytosine on the tRNA molecule, which is guanine. So our best answer here is A. All right, so what do you think is the amino acid W? It's arginine. Can you see why? This is a very common graphic in diploma exams. You can almost label every part of it. Notice structures 1, 2, and 3 are phosphates and sugars that make up the rails of these nucleotides. 4, 5, 6, 7, and 8 are the nitrogen bases. Now, can you see which ones are which? See, adenine, guanine, and cytosine are expected to be found on both DNA and RNA molecules. The nitrogen base on DNA and not RNA must be thymine. And the nitrogen base on RNA and not DNA must be uracil. So the answer to this question is A. Can you see why? Mutations occur due to a variety of reasons. DNA replication invites the most opportunities for mutations. Mutations that occur when where the genes have the potential to be extremely problematic. And in this course, we talk about the one type of mutation, the point mutation. Now, there are several types of point mutations. The insertion mutation, which is shown here, sees another cytosine nucleotide added to the molecules. And this causes what they call a frame shift mutation. It makes any attempt to read the triplets as codons later as futile. Likewise, this deletion mutation also causes a frame shift, meaning any attempt to read the triplets as codons later on would be futile. All frame shift mutations are also called nonsense mutations because the resulting sequence of triplets would reveal an amino acid translation that would produce a useless protein. Now, the substitution mutation switches out one nucleotide for another. Now, this could be a nonsense mutation, but could also cause a missense mutation, the production of a protein with an altered level of performance. This is another type of substitution mutation that sees a change to the DNA triplet. But since there can be several mRNA codons that translate to the same amino acid, this mutation doesn't change the amino acid, so the normal protein is transcribed. This is called a silent mutation. DNA is not limited to the nucleus. It's also found in the mitochondria and the chloroplasts of photosynthetic organisms. In this course, you're introduced to the hereditary patterns of mitochondrial DNA. There are genes on mitochondrial DNA that are transcribed and translated into proteins just like their nuclear counterparts. But unlike nuclear DNA, mitochondrial DNA is entirely maternal. Since the male only contributes the haploid nucleus of his sperm cell during fertilization, the haploid oocyte has all the organelles necessary to begin the life of a new zygote, including mitochondria and the DNA within. So this type of inheritance pattern has been used in confirming evolutionary patterns in sexually reproducing organisms, as well as the human out of Africa hypothesis. Males carrying a mitochondrial DNA mutation would have inherited this mutation from his mother's oocyte. DNA recombination technology has advanced in leaps and bounds over recent years, including the advent of CRISPR-Cas9 technology that refines gene editing to such a precise degree that it makes takes nearly all the guesswork out of the process. It's very difficult for a high school course to keep so up to date with all but the rudimentary processes of gene therapy. And it's described in such a way that you should be able to articulate the general process in its broadest terms. Now, this diagram, the desired gene is found in amphibian DNA. The restriction endonucleases act like molecular scissors to cut the gene out at a very specific location on the DNA strand. It does this by having the restriction enzyme locate a specific region of nucleotides that are on the boundaries of the gene in question. Now, once the gene is cut out, it's inserted using viral vectors into a bacteria that normally has no need for the product of this amphibian's gene. The bacterial gene has been exposed to the same restriction enzyme, opening it up to receive the new piece of DNA. Ligase enzymes, acting like molecular glue, inserts the new piece of DNA into the bacterial genome. However, the bacteria will unwittingly express this gene, producing copious amounts of this protein, and since it has no use for the protein, excrete it. Protein is collected and utilized as desired. Now, an extension of using restriction enzymes to cut up DNA is that of gel electrophoresis. By taking DNA from one person and exposing it to several different restriction endonucleases, cuts up the DNA into fragments. Now, because the non-coding regions of DNA differ from one person to the next, it's expected that the restriction fragments from two different people exposed to the same restriction enzymes will have different restriction fragment lengths. The differences in these lengths are shown using gel electrophoresis. The restriction fragments are added to a gel, and since DNA is a molecule with a charge, when a power source is added, the fragments will move through the gel toward the positive terminal of the power source. Smaller fragments moving more easily through the gel than the larger fragments. Now, the resulting bands on the completed gel between two different people will be aligned differently. The Hardy-Weinberg equation is formed from the idea that allele frequencies do not change from one generation to the next, and that a population will have a predictable portion of its members with specific genotypes for the gene being studied. Now, this Punnett square from your textbook shows the most possible genotypes in a population of mice given a gene with two alleles. The probability of allele big B appearing in this population is 70%, which means the probability of allele little b is 30%. Now, the symbol for the probability of the dominant allele big B as a number between 0 and 1 is small p. The symbol for the probability of the recessive allele little b as a number between 0 and 1 is small q. Therefore, p equals 0.7, q equals 0.3. The probability of two dominant alleles combining to form an offspring that is homozygous dominant uses the law of combining probabilities. That is, if the chance of a dominant allele appearing in an offspring is p, then the chance that two dominant alleles appearing in an offspring is going to be p times p, or p squared. The same process applies to the other genotypic combinations in the Punnett square. Now, the Hardy-Weinberg equation summarizes the Punnett square by saying that all the individuals that are homozygous dominant added to all those individuals homozygous recessive and added to all of those who are heterozygous for a given trait equals 100% of the population. Now, keep that in mind, because should the question be about incomplete dominance, then it doesn't matter which trait is p squared or q squared, as long as the 2pq is the blended trait. All right, so the upper equation here is in the data sheet. The one below it isn't, but it's a good to remember. All the dominant alleles added to all the recessive alleles add up to 100% of the alleles for that gene in this population. So in a population of randomly mating fruit flies, 93 have short wings, the recessive trait, and 473 have long wings, the dominant trait. So what is the frequency of the recessive allele? And what is the frequency of the heterozygous genotype? And how many long-winged flies are expected to be homozygous dominant? Now, I can do this, but the thing is, I was charging my pen and it's only, oh, it's 98% charged. Okay, I can do this then. All right, so first of all, when you get to a question like this, let's write out our what we have in the question. We start off with our equation. Oops. Hang on a second. All right. Just noticed I've actually changed some of the graphics on this thing. Okay. So 93 flies have short wings. Let's start by writing out the equation. It's my p squared plus 2pq plus q squared equals 1. I'll also put up the other equation, p plus q equals 1. Also, I'm going to reduce the size of this pen to something much smaller. All right. Now, these are raw numbers you see here. 93 have short wings, a recessive trait. So 93 out of our total population of 473 plus 93, that's going to give me a frequency of short-winged flies. So this is going to be 93 divided by now 473 plus 93, 566. So 93 divided by 566, and this is going to give us 0.1643. Now that is the frequency of the flies that have short wings. Now this q squared is the frequency of the homozygous recessive trait. In other words, the frequency of the flies with the short wings. First question is asking, what's the frequency of the recessive allele? The frequency of the recessive allele is q, but I have q squared. So what I can do is I can take this amount and simply square root it. So square root is 0.1643, and that comes to 0.405. That's interesting. How come I can't? There. So that's the end of the first one. The frequency of the recessive allele is 0.05. It's a frequency. It's a number expressed between 0 and 1. Now, if I'm going through this too quickly or you have any questions coming from this, please send me an email. I'll be happy to address these questions. All right, second part says, what's the frequency of the heterozygous genotype? Well, this term in the Hardy-Weinberg equation represents the frequency of the heterozygous genotype. Now I have Q, but I don't have P. But I do know, oops, you know what? I don't like the new Blackboard Ultra. All right, now I do have this equation. And if I can rejig this equation so it's in terms of P, so P equals 1 minus Q, and that is 1 minus 0.405. And I get 0.594. Now, this is what is really, really super important, and I can't highlight this the most. You're not going to get some nice even numbers that are easy to do in your head. You're going to get these odd numbers. And if you get these odd, strange numbers, when you find out square roots of these things and so on, make sure that in all your calculations, you are using the full number and you're not using a truncated, rounded number, because you will get wrong answers. So for example, this question here, I put 1 minus 0.405 and I get 0.594. Well, I've just calculated that from my calculator because it takes into account the big number, the whole number. All right. Now that I have P, my 2PQ, 2 times P, which is 0.594, times Q, 0.405. Now I'm writing the truncated number, but I'm keeping the full number in my calculator. So this comes to 0.482. And that's my answer for the second part of the question there. Right? It's expressed as a frequency, and if it's your final answer, then you can round it, to a. It doesn't say in this particular question, but usually you would have instructions on how to round it. Say round to 2 decimal places, for example, that kind of thing. The last question says, how many long-winged flies are expected to be homozygous dominant? Well, let's choose a different color. I shall use this one. This p squared is the frequency of the homozygous dominant genotype. So I'm going to need that one to start with. So p squared is my 0.594 squared which is 0.353. Like the full number, 353605202, but I'm just writing 353. Now that's the frequency of the homozygous dominant. The frequency of the homozygous dominant, the flies who have long wings. I'm not asked for frequency, I'm asked for how many flies. So to convert a frequency into a number of flies, I'm going to multiply this by the total number of flies, which is 566. And that comes to, well, 200 if I want to round this to the nearest whole number of flies. All right, so there you go. And you can expect questions in the exam that are that level of difficulty. Okay. Again, got any concerns, questions, please bring them to me. I'm happy to talk about this. Okay, there are 5 conditions the Hardy-Weinberg principle describe to keep these allele frequencies from changing in succeeding generations. Now, the first is that the population needs to be large enough to absorb the hazards of chance events so that a parasitic infection or an extreme cold snap, for example, doesn't kill off so many organisms that their allele frequencies are going to change. Now, that's called genetic drift. Now, there's a couple of types of genetic drift that you need to be aware of. First is the founder effect. This is a type of genetic drift caused by a small number of individuals separating from the original population and founding a new population. So the genetic diversity of the larger original population is going to be lost in the new population because the founders of the new population will carry only some, but not all, of the alleles of the original population. So that's one form of genetic drift. Another form of genetic drift is called the bottleneck effect. So this is another type of genetic drift caused by the near extinction events. So this slide illustrates the genetic diversity that cheetahs once enjoyed, but as a result of humans hunting these animals to near extinction about 100 years ago, the population has slowly built itself up, but only to a point where inbred lab mice have greater genetic diversity than populations of cheetahs left alive. And those alleles that added the extra diversity to the phenotype of these animals is lost forever. A second condition of the Hardy-Weinberg equilibrium is that organisms being studied not only reproduce sexually, but also randomly. Now that's to say each mouse in our picture here has an equal chance of reproductive success than the next mouse. So in this slide, we can see evidence that non-random mating, as the purple allele produces a favorable phenotype, and as such, later populations will show that allele increasing in frequency. Third condition of Hardy-Weinberg principle is that there's no mutations, or at least none that will change their biotic potentials or mutate one of the two alleles being studied into a third variety. See, this slide here shows we can see the allele from the ancestral population has undergone a mutation, and that results in a new allele appearing in the later population as another variant. Fourth, there are no organisms coming in from or populations carrying their own allele variants. So there are no organisms coming in from other populations carrying their own allelic variants of the gene being studied. Or you cannot have any organisms in our population allowed to leave, taking their alleles with them. This slide, we can see what happens to the gene pool of a population if some individuals leave, or if their seeds are carried in the wind to neighboring populations. The alleles that may be unique to the original population, they're going to leave, and that deprives later populations of that genetic diversity. And lastly, the genotypic frequencies remain unchanged if there's no natural selection against any of the phenotypes. So one mouse, for example, doesn't have a trait favoring or inhibiting survivability over the other mice. So this last part of the course, we talk about the interactions that occur between populations. And this is a fairly extensive chapter in your book, but I'll limit this summary to the important interspecific roles you're going to need to know for the exam. The first is the predator-prey relationship, and it seems one benefits while the other one suffers. But this extensively studied relationship between, say, for example, the snowshoe hare and the lynx reveals that perhaps the overhunting of snowshoe hares by the lynx reduces hare populations so much that the lynx populations are the ones that suffer. Mutualism, lots of examples in nature of mutualism. It's where both parties benefit from their interactions. The sea anemone here, the stinging tentacles of the sea anemone provide protection from the clownfish's predators, while the clownfish provide the sea anemone protection from its predators. Now, commensalism is a symbiotic relationship where one species benefits and the other one neither benefits nor is harmed. Now, some biologists actually argue that this type of relationship doesn't really exist because it's difficult to determine whether an organism is getting some benefit, albeit one that's very hard to see. Sharks, for example, don't seem to mind the suckerfish attached to their bodies, while the suckerfish receives protection from its predators and also bits of food discarded by the shark. And lastly, parasitism. This is a symbiotic relationship whereby one species benefits at the expense of another. It's extremely common in nature. Around 4,000 species of plants are parasites, and biologists estimate about 1 in 4 animal species are parasites, and that doesn't include the innumerable species of protozoa, fungi, bacteria, and viruses. So while the best known parasites are responsible for serious human diseases, most of them actually cause little significant harm to their hosts, which would ultimately harm the environment on which their own survivability relies. All right, so that's the Unit 6 summary. If there are any questions that stem from this summary, please give me drop me a line. I'll be quite happy to talk about anything, and good luck in your studies.